etc. Um, so, you consider the structure of a wheat kernel. I think obviously the structure. On one side, you have so-called the hairy structure, which serves to attach it to the main thing. And apart from that, when you consider the structure, you have the germ at the bottom, and this is what is removed during milling. The details we'll see later. And this germ is rich in oil. Fine. So you have the outer coat, which is rich in cellulose and hemicellulosic substances. And as you get inside, you have the endosperm, which is rich in starch. So, of course, I think the structure of any cell, plant cell. So you have the cell wall rich in cellulose, and the cell membrane rich in proteins and lipids. So that is how you get your, and starch and other things are present inside the cell. So that is how the structure is uh, found. Okay, just a minute. Somebody is disturbing. Okay. So I think the grain is usually between 5 and 9 millimeters in length and around 35 to 50 millimeters. As I already told you, you have a lot of layers. Basically, these layers are to protect the inside, and as usual, the outside of any of the substances found in nature is water resistance. You can see the waxy coating of most of the fruits or vegetables is because it prevents any moisture loss, etc. Similarly, in the case of the outer part of any stem is cellulosic kinase, which is again water resistant. So this is basically to prevent dehydration on one side, and on the other side, prevent any microbial attack. So these protect the seed by being waterproof and things like that. Fine. So you have the germ, as I said, which is rich in oil, and basically, in the case of milling, what is supposed to happen is that this hairy brush need to be removed. Okay, and the outer coat has to be removed. Normally, the outer coat is removed. Now, depending on whether it is what to say, refined flour or other things. The case of a refined flour or maida, this part is also removed. Now, ideally, when you go for any milling, right? Uh, Ideally, it should be that the wheat grain has to retain the structure, because what is basically happen, you pass it through rollers in a machine. So there are every chance that this structure can get cut anywhere. Okay, so but ideally only this has to be removed, and if at all the outer cover, the structure has to get you know what to say, intact as far as possible. So the brown is not wanted because of its color and texture. Color is one thing. Texture is because of the high content of cellulose, which does not absorb moisture. So you get a what is it? The final product texture, which is not that much desirable. And germ, as I already told you, it is rich in oil, and so when it's going to be stored for a longer period of time, it has a lesser shelf life. So that is the other thing. So the other thing which is important is the aileron layer, which is between the bran and the endosperm. So if you consider the structure, so this is the thing. This is your embryo or germ, as you call it. Okay. So the scutellum is also a layer between the germ and the endosperm. So bran and the endosperm, germ and the endosperm. Bran, the outer coat. Endosperm, one which is next to the what to say, the outer cover, and which is rich in starch. Okay. So the scutellum and the alluron both are rich in vitamins, so that also need to be retained. So when you go for milling, uh, it is better that first of all the structure need to be maintained. That is one thing. Second thing is that the preferably this germ need to be removed. So these are the main aim of willing. Fine. So prior to going to willing, again a brief history or the an overview of the uh, constituents. Of course, you have your cellulose in a higher concentration, and as usual, you have water first. Then you have the carbohydrates, which are present at a very high concentration, followed by proteins. So wheat actually has a good amount of proteins. Um, and the characteristic of this protein, of course, is gluten, which makes a elastic structure. Okay. Because of the higher content of protein, the wheat has a glassy or a vitreous look. So that is a shiny look, and this is said to be a hard wheat. Now, one of the method of classification of wheat, we'll see subsequently later. We have learned about the length, breadth, etc. That is one thing. That is whether they are very thin and slender or very strong or bold, as you call it. That is when it is fat, sort of. We call it as bold variety. And for example, most of our South Indian rices, we call it as a bold variety because they are short and fat. Whereas your basmati and other things, they are very thin and slender. That is the length is much more than the breadth. So this is called as a slender variety. So similarly, in the case of wheat also, one of the method of classification is based on the length to breadth ratio, as you can call. The second thing, of course, we'll see subsequently later, the weight of 100 kernels or 1000 kernels, etc. That we'll see subsequently. So this is basically the chemical constituents. As I already told you, the bran contains a small amount of fructans and resistant starch, but it is predominantly cellulose in nature. Now, when you consider this is the outer coat, here, if you see the alluron or other things, you can whatever you see is your cellulose, hemicelluloses, and lignin. Now, the characteristic feature of what is lignin? Lignin is um, form of cellulose which is linked to the phenols. Okay, so if you consider here all these things, you find that all the outer layer, your bran, alluron, etc., are rich in celluloses and hemicelluloses. Okay. So this is again basically to improve the thing. Whereas when you go inside, this is the inside. This is rich in starch. Okay. So that is a characteristic feature. So you have your hemicelluloses like arabinoxylans and galactans, which you would have studied in your second year, plus then vitamins and iron. And this is a germ which is rich in oil. So basically, again, as I said, during the milling, the entire structure except for this need to be retained. Okay, fine. So when you consider the protein and starch, if you consider the uh, outer layer, you find that the there is hardly any starch. Obviously, because the when you consider the biosynthesis of starch, I think I already told you is in the center. Why in the center of any fresh thing? This is because to germinate, you go from the center because from the outside you have a water resistant coating. So as the cell division take place from the inside, you have a rich amount of free sugars, starch, amino acids, etc. And whatever the polymers of amino acids, that is proteins, the polymer of um, carbohydrates, etc., they form the outer layer. So basically, when you consider the outer layer, okay, first of all, the starch is very less. The starch is normally found in the endosperm. Okay, and the protein is also found at a higher concentration. And if you consider here, only the endosperm has starch. When you consider the kernel weight also, that is, if I'm going to take one kernel, if you are assuming it is 1 gram, 82 grams of it comes from the endosperm. So endosperm is the widely present one. The next is your outer layer or a pericarp. Then the testa and the alluron and the scutellum, as you call it. So basically, your endosperm is the maximum uh, one present, and this endosperm contains all the starch which is absorbed. Of course, if you consider the embryo, embryo is your germ part where the amount of protein is comparatively less. Okay. Now, um, when after harvest, fine, when farmers, you know, send their loads of wheat grain. Now, this sampling is a procedure which is followed for wheat. It can also be followed for any of the methods. What is the significance of sampling? Like, when I'm getting tons and tons of wheat, I need to know the quality of wheat because the quality decides the cost of the material that comes inside. What are the quality factors? We'll subsequently learn. Okay. So when you take a quality factor, I need to take a certain amount of a sample for my analysis. Okay. Now, how do I take it in a very big drum? Okay. Um, if it is not properly mixed, you can have a sample which maybe have a high in moisture or any other thing. So to ensure that a proper representation of the menu is taken, what is normally done is on one side, if you want at a smaller level, I take part of it from the top, from the middle and the bottom. This is one of the methods of sampling. Or the other method which is normally followed, is mm, you're able to see the Jamboard or whiteboard. So
Yes, sir.
basically, in that sense, what happens is your wheat or rice or whichever it is, it is made as a heap. So this heap is then at the smaller level is made as a flattened out as a circle. In the lab, this is what is the thing followed. After this is divided into four parts. Okay, and what is done is say something like this part, and the opposite sides are taken for sampling. Okay, so now these two parts have got, so I get a smaller part. Right. Now, after this, what happens? The same procedure goes. Now, this small part is again made to a mount, then again flattened. Now, in this case, what happens? Now, the first time I had taken these two. Now, for the second time, what is done is these two parts are taken. Okay. After that, again they are taken out, again made to a mount. The third time here. So this is how the sampling is done. Now again for this, then normally say 1 kg or 2 kg of the sample is taken, and then finally whatever the representative sample might be something like you know a larger amount taken, and for the what to say monitoring purposes or other things, you take somewhere around 1 kg. Now, why is this near and maybe or a representative? Why I'm telling 1 kg or more? Now, to know the quality of each, a part of it goes for the analysis, like uh, the protein content, gluten content, etc., etc. Okay. Then other part of it has to be kept for reference. Why? At a later date, when you find, because this wheat is converted to flour, which then goes to various industries, either for manufacturing of biscuits or cakes or bread, etc. Now, under those conditions, then, if they find that there is some problem with a particular batch of bread or other things, normally it is a tendency to check whether there is a problem with the raw material. Now, when you check for the raw material, main raw material in these cases is wheat flour. So they go to check for wheat flour. Once they want to know what the quality of wheat flour is, then they'll come back to the main supplier. Now, now at the supplier's end or the company, for example, ITC or whichever it is, you have a lot of milling companies. They will have the sample kept of the whole wheat as also the ground flour. Why? To know whether there has been actually any what to say changes which have been observed, because at a particular initial stage you might not you might overlook some of the uh, discrepancies because it might not be significant, but then as time proceeds, we are going to take it for 15 days or 1 month. Problem is sometimes it can get aggravated. So that is how sampling is an important procedure for uh, most of the uh, companies. It need not be only for food companies. It can also have for a pharmaceuticals. It can be for. It's also for uh, every industry. Sampling is a must because uh, it gives an idea of how well is a particular thing made. Okay, uh, fine. So in the case of sampling, a larger basically when you are going to try doing it, uh, the grains can be have varying sizes, varying density and texture differences, because you have some when it is getting harvested, still you will be having fresh grains also. Fresh grains in the sense which are just born. Some might be 10 days old, 15 days old, etc. So ones which are 15 days or a month old will be more denser with a better texture, whereas those which are younger or immature will be very soft with the texture not being formed. The texture is mainly because of the protein content that is found, because initially you will have the starch in a very soft form. Later what happens is structural modification of the starch happens, so you get resistant starch, etc., which is not needed for you. So basically when I'm going to I have a gunny bag, when I drop uh, a full gunny bag through a chute, what is basically happens is that the larger size materials will be migrating away from the spout and they can form layers. So smaller ones can get inside, they will run faster. The larger ones will remain behind and outside. So the inner core will mostly consist of a smaller sized material, etc. So because of this, then for the purpose of sampling, we have what is called as a diverter type sampler, which is normally used. With the details we'll see subsequently. So this is installed at the end of a conveyor belt or in a spout. So what basically happens is that this again does the sampling. Whatever I have we have seen earlier, it is then cut into two halves and then subsequently uh, going for analysis. So uh, this this is how the things are. The details we'll see in a video. Right. So this is the sampler which is being used, the Bowden type of sampler. Okay. Okay. Just a minute.
Though many variables play a part in crop establishment, perhaps the most important is simply starting off right, with high-quality seed. But what determines the quality of seed, and how exactly do you test a sample? A good quality seed will have a high germination percentage. Germination refers to the number of seeds planted that are expected to sprout and grow. High germination usually results in good stand establishment. But germination isn't the only factor to keep in mind. Purity of the lot has a significant impact on crop establishment. Purity refers to the kinds of seeds and other matter present in a bag of seed. Ideally, you would want to plant seed from a source with a high pure seed percentage. Simply put, this means the lot has little inert matter and few weed and other crop seed, all of which could reduce stand and yield. Whether you plan to plant the seed yourself or to package and sell it, it's important to verify the quality of a lot of seed. This is where sampling and testing play very important roles. Let's consider 3 questions. How is the lot packaged? How much do you sample? How is the sample taken? Packaging differs according to seed kind as well as geographic location. Some seed kinds, clover or tall fescue for instance, are commonly sold in 20, 40, and 50-pound bags. In western Kentucky, soybeans and wheat are often available in 1,000 to 3,000-pound mini bulk bags or in bulk bins. And finally, there are vegetable or flower seeds which are typically packaged in small packs. These are treated differently because 1 or more full packets must be submitted to provide the minimum number of seeds needed for testing. In a traditional bagged product, we need to determine how much product to sample. The lab analysis will be based on the sample you submit, so you want to make certain your sample represents the lot. Each trier that is obtained from a bag or a bin is actually a subsample of the lot. Subsamples are combined in a single container at sampling, resulting in a composite sample. This composite sample is mixed and split down to a working amount for analysis by the seed laboratory. Through extensive research, a standard formula was developed to ascertain how much seed was a good representative sample. The general rule is 5 bags plus 10% of the lot. Here's how that works. For lots of 6 bags or less, each bag should be sampled and at least 5 trierfuls must be taken. For lots of more than 6 bags, 5 bags should be
So
sampled
this is
plus
the
10% of
procedure
the
to be
number of
followed for
bags in the lot.
sampling.
5 is the minimum number of triers required. For example, this is a carryover lot of red clover with 3 bags left. The amount of seed obtained from each bag should be equal to not bias the sample. In order to obtain the minimum of 5 triers and obtain an equal amount from each bag, you would sample each bag twice. When sampling the same bag more than once, be sure to follow a different path through the bag. Now let's consider a larger lot, one that contains 36 bags. With this lot, the sample will consist of 5 bags, plus 10% of the lot total of 36, which is 4 more bags. All 9 probes are randomly selected from the lot and are combined for the submitted sample. It is not necessary to sample more than 30 bags, so the minimum sample for any lot requires 5 probes and the maximum is 30 probes. Each sample should be taken randomly and evenly spaced throughout the lot. Samples should always come from unopened bags. Check each bag before probing to make sure it is the same kind and lot to prevent mixing. By following the sampling guidelines, you will usually acquire enough seed for testing. For example, with small seeded crops such as bentgrass, bluegrass, Alsike clover, or timothy, about 2 ounces is enough seed. For other small seeded crops like alfalfa, red clover, fescue, or lespedeza, about 5 ounces is enough. 1.5 pounds is sufficient for a crop like buckwheat or sorghum, and 2.5 pounds are enough for seeds the size of barley, soybeans, or corn. If you are only testing for germination, a minimum of 800 seeds is required. If additional tests will be run, such as Roundup tolerance or moisture, extra seed is necessary. Please note that with pelleted or coated seed, you may need to send additional seed as the coating material adds weight to the sample. If you have questions, the seed lab will tell you how much seed to submit. The type of seed not only determines the amount needed for testing, but also the method and equipment used to get the sample. Some seeds are more flowable than others, meaning they have a smooth seed coat and will flow into a trier very easily. Examples of flowable seed are clover, wheat, and crown vetch. Less flowable seeds are grasses, which have features on the seed that cause them to mat together. The least flowable seed kinds are native grasses, which are extremely chaffy. For sampling clovers and other small free-flowing seeds, a 30-inch trier with a ½ inch in diameter or a double tube sleeve type trier should be used. For the rougher cereal grains, use a trier that is at least 30 inches long and 7/8 of an inch in diameter, or a double tube sleeve trier. For other extremely chaffy, non-free-flowing seeds, the lot can be sampled by hand. The trier should be inserted in the corner of the bag and pushed diagonally across the bag. Triers must be inserted horizontally, though there are specially designed triers that don't require that method. Take care not to rip or tear the container when sampling, and be careful not to push the trier through the bag on the opposite side. Caution should be exercised to prevent damage to the seed. This is especially important when using a double-sleeve trier, which can crush the seed when being closed. All trierfuls should be approximately the same size, and remember to take a different path through the bag if it's necessary to take more than one trier from each bag sampled. When regulatory samples are taken, these same sampling procedures are followed. When an inspector samples a lot of bagged seed, the lot size is assessed, and the seed kind, variety, and lot number on the tag are verified. For example, this is a 160 bag lot of tall fescue. Determine the number of bags to be sampled and what equipment is appropriate for the seed kind. Randomly determine which bags you will sample. Based on the sampling guideline of 5 plus 10%, 21 bags should be sampled. Tall fescue is chaffy, so an AOAC trier will be the best tool for probing the bags. Considerations for bulk sampling are a bit different. Draw samples from random positions. If a sleeve trier is used vertically, it must be partitioned. In review, remember, an Indiana trier is used for small seeds. An AOAC trier is used for chaffy seeds or larger seeds. A grain probe is used for large quantities of large seed. Special circumstances, as in sampling extremely chaffy seeds like native grasses, may call for hand sampling. Sampling guidelines are available from members of AASCO, the Association of American Seed Control Officials. And finally, always remember safety when sampling seed. Be aware of your surroundings, especially any potential hazards, including equipment operators, low clearance areas, and exposure to treated seed. Take necessary precautions, which may include wearing a bump hat, steel-toed shoes, and latex gloves. On the sample bag, please include the seed kind and variety, lot number, and if the seed is treated. Seal the samples into a sturdy bag or box and send to a testing facility. When having seed tested, be certain the lab is an AOSA-approved lab. This ensures the lab meets certain qualifications in knowledge and proficiency. At the lab, sample information, including the seed kind and variety, lot number, treatments, as well as other data provided at submission, is logged into a database. Samples are divided into groups, those that require purity evaluations and those that require germination only. Samples that require a complete analysis go to the purity lab prior to the germination lab. Germination-only samples go directly to the germ lab. Samples are mixed prior to evaluation to eliminate any settling or segregation that may have occurred in transit. This is especially important with mixes that may have different size and weight seed. Samples that have a purity evaluation are identified, mixed, and divided into a working weight. This ensures that the seeds being examined are typically of the submitted sample, which, if it has been sampled properly, is typical of the lot. Working weights are specific to seed kinds. The guidelines are published by the Association of Official Seed Analysts in the AOSA rules for testing seeds. The working weight has two parts, the purity portion and the noxious weed portion. The purity portion is physically separated into pure seed, inert matter, weed seed, and other crop seed. Any noxious weeds in this portion are also identified and counted. The noxious weed portion, which is typically 10 times that of the purity portion, is examined for noxious weeds only. Noxious weeds in Kentucky include all seed kinds found in the purity portion are identified, then weighed to determine a number per pound for the seed lot that was sampled. The pure seed portion is packaged and sent to the germination lab for germination testing. Over in the germination laboratory, seeds are planted and placed in germination chambers according to strict rules. These rules deal with the number of seeds planted, temperature, light requirements, and any other special instructions. At various times, samples are evaluated for sprouting and normal seedling development. At the final count, the total germination is recorded and a germination percentage is determined for that seed lot. The purity and germination information is reported to the submitter. These reports are important as they can be used to verify quality of a newly received seed lot, verify quality of carryover or stored seed, and used to generate a new or replacement seed label. We hope you've enjoyed our program. In the seed industry, the importance of a representative sample and the value of testing cannot be overstated. Neither can the use of proper
reduces the size of the Borner divider reduces the size of a grain sample while maintaining the representativeness of the original sample. Each pass of the sample through the divider splits the sample into two equal representative portions. When operating the Borner divider, check the divider for condition and cleanliness. Place empty collection pans under the discharge spout. Close the hopper valve. Pour the sample into the hopper. Open the valve quickly. For larger samples, feed more grain into the hopper during the dividing process.
This is for dividing or sampling rather. I say whatever I was telling manually, like making it as a mound and then flattening it. So this is how it is done. This mixing is to ensure that you get the right quality of the material, because as I already told you, there are chances that the bigger size can settle down and the smaller size can settle down. So this is repeated. This is called as a Bohner divider, which is used for um, wheat sampling actually. So this is your sampling procedure. So for example, if you're taking say 1 kg, at the end of the day, uh, or a 2 kg, you get around 500 grams after repeated sampling and dividing. So that's all for today. Um, let's have the attendance.